Good morning, students. In our previous class, we studied about the Griffith experiments. In this Griffith experiments, Griffith searched for the genetic material, but at the end of the time, Griffith gave a conclusion. One of the transforming principle modify the R strains into S strain. Okay, but they did not give any explanation or the biochemical nature of that substance. Okay, only he has said transforming principle can cause the modification in R strain. He did not give any explanation on biochemical nature of that chemical substance. Later, Griffiths. Let's our discussion starts with another experiment, that is biochemical characterization of transforming principle. Biochemical characterization of transforming principle. This experiment did by Oswald Avery, Colin MacLeod, McCarthy in 1933 to 44. In some books it is also 34. Oswald Avery, Colin MacLeod, McCarthy. They did or they repeat the experiment of Griffith. Okay, they repeat the experiment of Griffith and at last gave a conclusion of transforming principle. Okay, of the biochemical characterization of this transforming principle. For that, again, they used heat-killed S strains. They used heat-killed S strains. From this heat-killed S strain, they extract three substances. Okay, those three substances are the protein, RNA and DNA. Okay, so when they extract these substances from heat-killed S strain, these three substances separately allowed to infect these R strains. Okay, this is the R strain bacteria, this one also R strain bacteria. In one culture, they insert these protein substances. Whenever they insert the protein substances, it didn't find any transformation of R strain. There is no transformation. In the next step, they inject this RNA into R strain. Again, there is no transformation. R strain will be in the form of R strain only. At last, they inject this DNA into this R strain, then there is a transformation occurs. That means, in these three substances, there is an doubt. Okay, in prior, what the scientists thought actually either protein or the DNA is the genetic material accurately. Okay. So for this, there is a biochemical characterization of the transforming principle. They use three types of substances for these R strains. When they use the DNA, then there is a transformation in R strains. These R strains now converts to the S strains or the disease causing strains. Okay, based on this, they gave a conclusion. DNA is the accurate genetic material. Protein is not a genetic material. Okay, so DNA is the accurate genetic material. They also did experiment by using some enzymes. Okay, they use enzymes because of DNA only cause this transformation or there is any combination. For that, again, they use some digesting enzymes. When they use protease enzyme, this protease enzyme digests the proteins actually. So when there is a digestion of proteins, okay, when there is a digestion of proteins, almost there is no result actually, the transformation happens. Okay, the transformation happens. When there is a digestion of RNAs, by using RNAs, the transformation happens. But they use DNAs. Whenever they use the DNAs, automatically there is an inhibition of transformation. That means DNAs digest the DNA. Whenever there is a digestion of DNA, automatically the transformation will be inhibited. The R strain will not be modified into S strain. So based on this, DNA will only cause the transformation in this organism or in this strain. So that's why they give a conclusion, DNA is the accurate genetic material. Okay, in this, DNA is the accurate genetic material. But most of the scientists were not satisfied with this result at that time. Okay, so then there is another experiment. Actually, this is a particular perfect experiment, but most of the scientists were not satisfied with this result at that time. Okay, now there is a final proof. This is the unequivocal proof. That means without no doubt and debates, the accurate genetic material is the DNA at last. Okay, in 1952, there is an important experiment. By this experiment, in organisms, the DNA is the putative or the universal genetic material. So for that, they did an experiment. That experiment, just you see, done by Alfred Hershey Martha Chase in 1952. So for this experiment, they use two organisms. One is the bacteria and the second one is bacteriophage. These are the experimental materials. So there is an bacteria, there is an bacteriophage. So bacteriophage means what? Virus. Okay. So these viruses are the unique biological entities infect every type of cell actually. So nowadays you clearly observe the COVID-19 is a very, very dangerous virus. Okay. Then you see this is the virus. This virus infects the bacteria. Okay. When the virus attacks on bacteria, that virus we are called as bacteriophage. Okay, if the virus attacks on bacteria, that virus we are called as bacteriophage. Here phage means particle of the virus. So in this experiment, they use bacteriophage and bacteria. At the same time, they use radioactive isotopes. Okay, radioisotopes, that is sulfur 35 and phosphorus 32. These are the labeled isotopes or the radioactive isotopes. Okay, so this virus, first of all, they grew in a culture or in a medium which is containing, that is sulfur 35. Okay, so when the virus grow in a culture or in a radioactive isotope, that is S35, okay, this is the medium only for this virus. So after few generations, what they observed actually in this virus, there is a radioactive sulfur which is present in a protein coat. Actually in the virus there are two important chemical substances. With these two chemical substances only the virus will be made. Okay, so just you see, this is the virus when they grew in a medium that is sulfur containing, this is the radioactive sulfur. After few generations, they observed this virus contains radioactive isotope in protein coat only, not in nucleic acid. Okay, this S35 is present only in protein coat because of in the protein synthesis or in protein formation there is an sulfur present. Okay, there is an sulfur. Okay, the sulfur is present only in proteins. That's why in the protein coat only they absorb this radioactive isotope. Then there is some viruses. These viruses they grow in another isotope containing media, that is radioactive phosphorus. Okay, that is radioactive phosphorus. So whenever they grow the virus in radioactive phosphorus, after some time or after few generations, what they observed actually in this virus, there is an nucleic acid. This nucleic acid is the radioactive nucleus. That means the phosphorus is present in DNA. That's why the phosphorus is incorporated into this nucleic acid, not in a protein coat. Now there are two different viruses, just you see. Okay, this virus is the too dangerous actually. So virus means a poisonous substance. There is no use of these viruses actually. Nowadays you see the COVID-19 is a too much dangerous viruses. There is no vaccine still now because of there are some lot of modifications, lot of mutations. Okay, so just we think later. Now these are the two viruses actually. So one is the radioactive sulfur containing protein coat virus, and the second one is the radioactive DNA. Okay, not protein coat, radioactive DNA containing virus. So these are the two different viruses. These two different viruses allowed to infect separately on bacterium. Okay, these two viruses, first one is the sulfur containing protein coat virus, and the second one phosphorus. This is a radioactive or the labeled, okay, phosphorus in the DNA. Because of in DNA, just we observe the phosphorus. Okay, this radioactive sulfur containing, radioactive phosphorus containing viruses allowed to infect separately on the bacteria. So the first step is the infection. So now one thing you remember, when the virus attacks on bacteria, only the genetic material is inserted into the bacteria. Only the genetic material inserted into the bacteria, the protein coat remains outside. Okay, whenever the protein coat remains outside, this is called as ghost. Okay, ghost. Only the genetic material inserted into this bacteria, the protein coat remains outside. Now you see clearly, after infection, there is a process, blending process. That means in this blending process, we separate the protein coats from the bacteria. Okay, there is a separation of virus protein particles from this bacteria in blending process. Okay, so after blending process, the viral genetic material insert in these two bacteria, they multiply rapidly and form number of virions. Okay, then there is a centrifugation mechanism. In this centrifugation mechanism, they absorb the viruses and the bacteria. Okay, the viruses are lightweight actually, they absorb in supernatant form, in sedimentation form, they absorb the bacteria. So in this is the first experiment, this is the second. Okay, these are the two. So when the sulfur containing protein coat virus attacks on this bacteria, whatever the variants are formed in this bacteria, there is no sulfur containing protein coat. These are the normal viruses. Because of the sulfur coat containing protein just will remain outside of the host. Okay, that's why this protein coat remains outside. In this variants, we can't find any radioactive conditions in these variants. Okay, now in this you observe, so this is also form number of variants actually like this. So in this, whatever the variants are formed, the variants are the radioactive DNA containing viruses. Okay, radioactive containing DNA viruses. So clearly you observe the protein coat containing S35 sulfur, that is a radioactive sulfur. So this is a radioactive phosphorus which is present in DNA. So this protein not enters into this, that's why whatever the variants are formed, those are normal viruses. Okay, the DNA inserted into the host organism, so then this is the radioactive DNA, that's why the variants are formed which is containing the radioactive DNA containing viruses. So based on this, the accurate genetic material is the DNA, the putative genetic material is the DNA. So this is the final conclusion of the final experiments. So in this you clearly observe, first step is the infection, second one is the removal of protein coats by blending, and the third one is the centrifugation. In the centrifugation process also we separate the bacteria and viruses. So then there is a conclusion, the accurate genetic material is the DNA. So one thing you remember, so there is a radioactive condition, that is S35P32, number of times was the question. Okay, S35P32, there is a bacteriophage, there is a bacteria, the scientists are Hershey and Chase, the year 1952, there is an unequivocal proof, the accurate genetic material is the DNA. Okay, so these are the experiments. First experiment is the Alfred Griffith experiment, and the second one is the biochemical characterization of the transforming principle, that is Avery, MacLeod, McCarthy. Okay, then the third experiment is that is Hershey and Chase experiment. This is the final proof. Okay, Now the genetic material is the DNA. We go for the some characters of this DNA and other genetic material also. There is an RNA. Now just we compare DNA and RNA. So if the substance act as a genetic material, some properties they have, so those properties we will discuss. Okay, next, after unequivocal proof, properties of genetic material, means there are two important genetic materials actually, DNA and RNA. That means DNA versus RNA. If one chemical substance, okay, these are the two chemical substances. If two chemical substances have some properties, that means if one chemical substance act as a genetic material, should follow the some criteria. So what are those criteria? Just you see, it should be able to generate its replica. That means it should have the ability to generate its replica. So already we know there is an complementary base pairing. There is an template manner stance, okay, complementary base pairing template stance. So these two, DNA and RNA, actually genetic materials, but in these two, the DNA have the capability, more capability, both are undergoing replication, but the DNA is a double stranded, RNA is a single stranded nucleic acids. Okay, in majority of the organism, DNA is a genetic material. But what are going, so what are we discussing actually? The properties, if one chemical substance act as a genetic material, some of the criteria should follow. Okay, this criteria should follow DNA and RNA, but this DNA is the double stranded, RNA is the single stranded, there is a complementary base pairing rule, okay, there is a template strand manner. Now, it must be expressed in the form of Mendelian characters. So, what Mendel said actually, the characters are controlled by factors. So, with these factors, the characters are transferred from one generation to other generation. This is the factor. Now, it should be stable both structurally and chemically. It should be stable both structurally and chemically, and it should go for slow changes. Changes are necessary, but it should go for slow changes. Okay, these are the criteria of the chemical substances. In these two, which one follow this criteria? Okay, first property, almost there is a replication mechanism. So now it should be expressed in the form of Mendelian characters. Then we go for the structure of the DNA and RNA. Compared to the RNA, the DNA is more stable, chemically less reactive actually. RNA is the less stable, more reactive. Just you see structural variations in these two. In pentose sugar, in RNA, at the second position there is an highly reactive, that is hydroxyl group. But in DNA, in the deoxyribose sugar, there is no reactive hydroxyl group, then there is a removal of oxygen, only hydrogen is present. So this adaptation gives the stability to the DNA. Okay, and the second thing, structurally there is a variation between the nitrogen bases also. In RNA there is an uracil, in DNA there is an thymine. Okay, chemically it is called as 2,4-dioxy-5-methylpyrimidine, that is 2,4-dioxypyrimidine. There is no methyl group. So by this methyl group also, the DNA is more stable compared with the RNA. So once you just recap the Griffith experiments, in this Griffith experiments, the Griffiths killed the S strains by heating. So whenever he killed the strains by heating, those heat killed S strains combined with the R strains, again the R strains are converted to S strains. One thing you observe clearly here, the heating, whenever we destroy the heat killed S strains by heating, again we can't destroy complete properties of that S strain. Some of the chemical substance will not be destroyed it will be transposed into R strain and converted to S strain. So that's also stability of the DNA. Okay, so actually there are two strands in the DNA. These two strands are bound with hydrogen bonds. So when there is a heating, just there is a denaturation or the separation of these strands. Okay, these separated strands just adapted to cool places. Again, there is a binding of this DNA strands. Okay, based on this, the DNA is the more stable compared with the RNA. So then, in these two genetic materials, the DNA go for slow changes. Okay, if one chemical should undergo slow changes, that means every time in life cycle it will not be changed. Okay, it will not be changed. So in RNA, it goes for fast mutations. By this structure, the RNA goes for fast mutations. So by this, the RNA was not stable compared with the DNA. So in this variations, in these two chemical substances, the DNA is the more stable. Okay, that means the DNA store the genetic information, RNA transfer the genetic information. Because of, you see, central dogma of molecular biology, the stored information passed to the mRNA. Now the information carried out by mRNA to the protein. Okay, that means RNA directly linked with the protein synthesis, not the DNA. The DNA is a reserve bank, like reserve bank. Okay, it is the chemical basis of heredity. Store the information passed by this RNA. Okay, that means for the storage there is an DNA, for the transmission there is an RNA. Okay. So according to central dogma of molecular biology, so this RNA again having some mechanisms. So whatever the metabolic activities are there, those metabolic activities are completely surrounds to this RNA only. Okay, so there is a translation, there is a splicing, there is a metabolic activities, all these around this RNA only, not to the DNA. That means in these two genetic materials, RNA is the first genetic material because of all the metabolic activities, translation, splicing, all this around this RNA only. Okay, RNA only transmit the genetic information, DNA store the information. So by this, sometimes RNA act as a catalyst, that is 23sRNA ribozyme. So by this, RNA is the less stable, but the surrounding metabolic activities around this RNA only. Okay, so from ancient times when we observed, in this DNA and RNA is the first genetic material actually, most of the activities around this RNA only. Okay, so according to the evolutionary mechanism, this RNA modified into DNA. Okay, structurally they adopt the stability. Okay, this RNA modified into DNA, but first genetic material is the RNA only. So from this RNA only, there is some modification. Okay, the single stranded converts to double stranded. There is a structural variation. So that's why which one is the first genetic material? Actually, RNA is the first genetic material only, but the stable genetic material is what? DNA is the stable genetic material. So this is the double stranded genetic material. It is the stored information will be stored in the DNA only. Okay, so this is about properties of genetic material and DNA versus RNA and RNA world. Okay, so tomorrow we'll start our discussion with DNA replication. Okay.